Hello and welcome to Psyched, the show where we explore psychedelics through social, economic, and political perspectives. We have Dana Beal. Dana Beal, heir to W.M. Burroughs and Allen Ginsberg, who reinitiated ibogaine development with Howard Lotsof in the December of 1980, is the principal champion of Robert Gouterel's hypothesis identifying ibogaine mechanism with the neuro-plasticity of REM sleep, as opposed to the NIDA's reduction of ibogaine effect to blockade of a single alpha-3 beta-4 nicotinic acetyl-choline receptor. In the IBOGAINE Story report on the Staten Island Project, Beal was the first to correlate ibogaine visualizations with the Penrose Hameroff hypothesis of a photonic basis of consciousness in the microtubules of neurons, correctly intuiting that the neuroplastic function neurotrophins play in correcting long-term memory, heralded the findings of Ron in 2005-6 of a role for nerve growth factors such as GDNF and ibogaine's persistent effect. In 2014, that led him to predict correctly that ibogaine would be effective for Parkinson's disease. In February 2016, he issued an impassioned call during UNGASS for the countries of the world not to reflexively schedule ibogaine in the midst of a world opiate methamphetamine crisis on account of the US ban. Dana, thank you so much for joining us and welcome to Psyched.
Unmute. Hello.
Hey, we can hear you.
I'm trying to get the share screen here.
You can also turn on your video if you'd like.
Start my video. Sorry. Okay. Hi. Now I want to share screen. Share screen.
Share.
Anyway. There we go. This is the one I want here. There we go. So, yeah, I actually has something to do with starting ibogaine. There was a certain point, Ronald Reagan had just been elected, and we realized that marijuana wasn't going anywhere soon. We said, well, let's go back to the drawing board. And we had this thing called ibogaine. We were having a big problem with heroin at the time. And we said, let's develop ibogaine. And we had no idea what was involved. It was like a 10-year process. And about 10 years later, we brought it to the National Institute on Drug Abuse. And ibogaine is the only drug ever studied by the government that is an illegal psychedelic drug for treatment of drug addiction. And our indication was actually cocaine, for which they had nothing. And in 1996, through a sleight of hand, there was a panel that decided not to proceed with ibogaine development with NIDA. And on that panel was Suboxone. The Suboxone manufacturers got themselves onto the panel. So today, Suboxone is the only thing that you can take for drug addiction. And then Rick and Benkheiser is on their way to repeating the success of the Sacklers. But the interesting thing was ibogaine gave birth to another drug, which was a NIDA drug called 18-MC. And 18-MC is being currently promoted by a group called MindMed. So our first slide here, you can see this is the scaffold of ibogaine, and methcoronaridine down here at the bottom, 18-methoxycoronaridine, has, you see, all these other things are kind of like less bulky, but this has this extra carbon on it. And so this makes 18-MC more similar to volcangine, which is another psychedelically inert drug. And why did they come up with this? Well, they found in animals it got them to stop doing drugs, and they were trying to find a non-psychedelic alternative to ibogaine. So that was really what it was about. And that's somewhat got overlooked in the enthusiasm for the psychedelic rush. Anyway, I have to go through these slides very quickly, but this is the dopamine pathway that people are pretty much familiar with two neurotransmitters, serotonin, which is the basis of like psychedelic drugs, a lot of them, and dopamine, which is the thing that makes you jump if you're confronted with food, sex, water, or drugs. And actually, there's a little experiment that was done. And you see that they gave a rat juice, and the rat registered it and imprinted it, and there was a dopamine increase. And when they started giving the juice to the rat, they started flashing a light before the juice. And lo and behold, it moved. In other words, here's before the juice. The juice is still at the same place, but the animal alerts prior to the reward when it gets that stimulus that it's going to get a reward. So all of, like life is based on being able to animals being able to eat. So, and procreate. So without that stuff, you couldn't function, but you see there's a problem with it. Here's the pathway, by the way. This is dopamine, which comes from L-dopa, and is then converted into norepinephrine, so that the same pathway is used either for rewarding stuff or fight or flight. But basically, you get the attention of the animal in a hurry. So this is very interesting because you don't usually think of your central nervous system as being outside your brain, but we have a whole really important part of the central nervous system on top of our kidneys. I'm going to talk a little bit later about glia-derived neurotrophic factor, which is closely related to dopamine. It's one of the nerve growth factors that ibogaine expresses. And it is interesting that mice, where they knocked out the nerve growth factor gene, were born without kidneys. They not only didn't have adrenal glands, they didn't have kidneys. So they didn't live very long. But here you got like a rat having sex, is a male rat. And he starts over here and he starts feeling good, and then he gets aroused, right? And then he does it for a while more, it feels really good, and he has an orgasm, and then they drift off to sleep. Okay, the important thing to understand about dopamine is, Nancy Reagan used to tell us it's the pleasure switch. It's not. It's the arousal switch. But if you think about sex, if you can't get it up, or if the lady is just not interested, nothing much happens. So, this is all intimately related to sex. So, it's attached to your kidney. Anyway, how do drugs come into the picture? Well, either like over here you have cocaine and Ritalin, and over here you have methamphetamine. Methamphetamine is a releaser, cocaine and Ritalin block reuptake, but you get a flash of dopamine in like the synapse, and if it's a really strong hit of methamphetamine, you get a situation where it kind of blinds the receptors. It's like too much light at once. And some of your receptors at that point go into hiding. They actually retract. There's this thing called beta-endorphin, not beta-endorphin, beta-arrestin, excuse me, beta-arrestin, which kicks in so that, you know, you don't have excitotoxicity. And see, the excitotoxicity is a real problem. This is the effect of methamphetamine in mouse midbrains. And you see that after the meth, just one time, there's less of these bodies and there's less of this tracery of dendrites. So you're literally having burnout. Now, back at the beginning of time, in 1993, roughly, there was this split in the ibogaine world between those who thought that the visualizations do something and people who said, no, we can use this thing called noribogaine, and that was Deborah Mash, which is desmethylated ibogaine. It's just ibogaine after it goes to your liver. But it actually is a very active metabolite, and it doesn't cause visualizations. It is actually, with ibogaine, you go through visualizations, deep concentration, and then insomnia. And it lasts about 36 hours, and nobody wants to ever do it again unless they get in the unfortunate position of being addicted to heroin again, in which case you end up doing it again. So the point is, at that point, when this debate was taking place, I encountered this quantum coherence in microtubules. And what it said was that the brain is like a giant fiber optical switch, and that you have these microtubules exactly 1 nanometer wide, 1/1000th of a meter, and they spontaneously laze so that your brain is like a gigantic Bose-Einstein concentrator or a laser, and it is based, mentation is based on light. Well, if mentation is based on light, then maybe the visualizations do something. And so I was kind of a proponent of the visualizations do something because that's what everybody told me. And after all, I'm just a journalist. So I said, this is going to be the key, like a key clue. And then I printed my book and we didn't go any further. But right after that, I published a book called The Ibogaine Story. There was another woman who came out with some stuff that comes right after Hameroff and Penrose, quantum coherence of microtubules. There was a woman named Nancy J. Woolfe. It was on a basis of memory storage in animals. And what she said was that you take short-term memories and convert them into long-term memories, and they're like, you know, snapshots. And they're embedded in the microtubules, and you access distributed memories that are embedded in calcium. Now, when I died, and I was dead for 3.5 minutes, they had to freeze me. So I don't remember what happened, not only during the time I was in coma, but for the whole day before that, because those memories are lost. They were not encoded in calcium. So I'm pretty sure this actually works. And what they show is, this is a very important slide, because if you look right here at the second part of it, right, you don't have to go far. You find out that neurotrophins, which are like nerve growth factors, are involved in ordinary revision of memory, right? In other words, say you got, your wallet was stolen and you couldn't figure out who did it at first and you suspected the wrong person. But then later you find out, no, it was somebody else. Well, then you're going to go back and correct all the memories a little bit to reflect what really happened. And this is done because you actually abolish memories and make new ones, except they're not really, they're more like edits, you know. But this is very important in addiction because you got to do something to change memory if you're going to change addiction, because addiction is lodged in memory. Okay, so in about year 2000, by the year 2000, before they discovered the growth factor stuff, they had this basic understanding of ibogaine. And people kept coming up with ways some combination of short-acting effect might be producing a long-acting effect. And the first thing they found out was that ibogaine is a kappa opiate agonist like Salvia. And if anybody has ever taken Salvia, I think there's probably some people here who've taken Salvia, it's a dysphoric hallucinogen, and it's not very pleasant. But it does a lot of really useful stuff biologically because it's modulating mu, and it's keeping you from overdosing on mu, but also it makes mu work after mu should stop working, say if you had an accident. It inhibits cyclic AMP, which is there's this stuff called adenylate cyclase, which is implicated directly in the withdrawal, and ibogaine gets rid of that. And it inhibits beta arrestin, so that you're bringing back receptors. Now, this is the very guilty nicotinic acetyl-choline receptor, the alpha-3 beta-4. And this is the same in ibogaine and 18-MC. So what they did was they came along and said, this one thing is basically doing the whole thing because it's the same. And the animals are ceasing to use drugs, and the receptor is the same. And that was basically, they were reductionists. They didn't believe in entourage effect. Okay, now this is the most important thing. It took it a super long time to understand, but Mash came up with this interesting finding with her noribogaine, which is that noribogaine blocks the serotonin transporter like a Prozac or cocaine does, but in a different conformation, which means here's the standard one, right? For cocaine, it's open on the top. Here, it's closed on the bottom, and it's internalizing. And this is the only, ibogaine is the only thing that does it. So it was kind of a mystery how it was happening, but it was like a sticky Prozac. That is, it hung around for a while. This is sigma 2. Sigma 2, actually, you do not want. It is the same as the stuff that you get from thorazine, and it causes tardive dyskinesia if you have too much of it. And the only good thing about it is it blocks NMDA toxicity, which you don't particularly want because it's like glutamate toxicity, it's inflammation. And that is this, see? This is, we thought when we found it, this explains ibogaine effects, because what does it do? It's activated by ketamine. It cuts opiate withdrawal and supersensitization to stimulants, which is the thing where you start doing coke and you can't stop. And it also may act to decondition the M5 glutamate pathway, on which there's a little bit more later. But I was looking at this in the year 2000. I was giving a speech in a presentation on this, early presentation of this. And I said, you need a way that calcium can get in to the cell without causing a stroke. Because you see what happens with this, if too many glutamates hit this, when you have a stroke, the little clot starts killing cells, and they freak out and they start sending out glutamates, and you get a glutamate cascade, and the stroke can spread over half the brain. So you got to come along and you got to plug this. And what I was saying, well, you also have to let it in to rebuild the stuff from addiction. And what this did was this posited a growth factor. But what these other people were saying was, it's kind of complicated. This is the non-GDNF explanation of how many temporary effects can cause a permanent effect. And it doesn't really hang together, because what they're saying is that acetyl-choline is coming along and dampening dopamine. And this thing, somehow you're dampening the dopamine system. You don't want to dampen the dopamine system. You want to decondition it, but you want it to work just fine. Anyway, this is the big breakthrough from 2003, 2005. And as you can see, these are neuroblastomas. And when you give them GDNF, which ibogaine increases 12-fold within 6 hours, they start growing and they develop a network of dendrites. So you're literally invigorating the dopamine system. You're re-sprouting the dopamines, and that's caused by GDNF. So GDNF ordinarily goes through the edge of the cell, comes along for a receptor. It as a companion receptor underneath the membrane, and you have a cascade down to the nucleus, and some messenger RNA comes out. Messenger RNA goes through a bunch of like little sacs that vesicles make it into a finished GDNF, and the GDNF then goes out. See, this is the 12-fold increase, but that spike turns on this machinery where you start making GDNF all over the place. This is like the Sorcerer's Apprentice with the, Mickey Mouse with the buckets and the mops. Pretty soon you have GDNF going, regenerating the entire system, and you don't need any ibogaine at all. But you actually have ibogaine, and this is the important thing. They finally found out, and this is the most recent thing, that the stuff actually gets inside, right? Ibogaine is actually getting into the transporter. Well, when it gets into the transporter, then you have an explanation for this question mark. How is it doing it? Well, it's getting into the transporter, and the transporter is, on a terminal cell, and you see this terminal cell is making dopamine and serotonin over here. It can make also all kinds of nerve growth factors and steroids and all kinds of other stuff. So this is a final point here. This is the M5 glutamate receptor, and this is the pot receptor, right? And you notice pot is a backwards signaling neurotransmitter. It's telling the glutamate that it's firing too much and telling it to calm down, chill out. Well, ibogaine is actually working the same way as exogenous pot, coming along to the terminal, right? Going into the transporter, which is there to suck serotonin back in, jamming the serotonin transporter so that you have more serotonin left over in between the synapses of cells. And lo and behold, it turned out that noribogaine, but not 18-MC, exhibited similar actions as ibogaine on GDNF expression and ethanol self-administration. So, oops, there's something missing with this Brave New World rational drug development compound, which is it's not turning on the nerve growth factors. Oops. And here is the other side of it. 18-MC does not go into the herd channel, and ibogaine does. It's just a slippery little molecule, or noribogaine. And that is a calcium channel that you don't want to block. Remember chloroquine? You know why they really don't like chloroquine? Because it's an arrhythmogenic. And they're trying to ban all arrhythmogenics. So right now, we can't even get ibogaine into trials. A dose too low to cause arrhythmias, like 4 milligrams per kilogram, which does happen to work really well for crystal meth craving and for Parkinson's. But we can't even use it because it might do something, well, it definitely does do something at a higher dose. It might do something in the lower dose, but we can't tell because they won't let us do the trial. Anyway, this stuff is off to the races. I think the important thing is, you see with methamphetamine, 18-MC is not as effective as it is for nicotine. Nicotine is really effective, and J.R. Ron may have a great smoking cessation tool there. But you see, like I said, and I'm going to, in a minute, go into how ibogaine works, but they're saying it's all in one place, one receptor in the middle of the brain called habenula, the interpeduncular habenula, and that if you can just zap, some nicotinic receptors in the habenula, you solve addiction. Now, the problem with that is, you see, this is Wellbutrin. You guys will know Wellbutrin. And Wellbutrin, what this shows is Wellbutrin doesn't work. It's less effective than placebo. When I was in jail, there was a guy crushing up his Wellbutrin and snorting it like Coke. You figure it out. But 18-MC has got some pretty great, like, mouse. Again, there's that like nicotine thing. They should study it for nicotine. I don't think it's going to be that great for like speed is my hunch. But the question is this a side effect or a mechanism of action? And I want to read this whole thing, even though I know I don't have it forever. This is Stanley Glick, the inventor of 18-MC. The hallucinations are just an unfortunate side effect, Glick asserts, explaining that ibogaine works on the brain like a hybrid of PCP and LSD. Part of the problem is that when you go through this thing, it's so profound, you've got to believe it's doing something. In part, it's an attempt by the person who's undergoing it to make sense of the whole thing. Generally speaking, Glick's research in rats has shown the ibogaine dampens the brain's so-called reward pathway, reducing the release of neurotransmitters like dopamine, which cause the highs associated with everything from heroin to sugary foods. The compound has also been proven to increase production of GDNF, a type of protein that quells cravings, and to block the brain's nicotinic receptors, the same spots that are stimulated by tobacco and other addictive substances. Now, nobody told the writer, and Glick is on that paper, oh, by the way, we don't do that. We don't actually do the nerve growth factor at all. We're just doing this one thing. But the other thing is, I just, do a thought experiment here. Imagine that you're recording a symphony orchestra. You're not quite getting the sound right. Somebody comes in and says, double the number of microphones in the orchestra. Well, to begin with, you could turn down the gain, but you'd also have more granular, better sound. So you don't want to have to like amp up the brain with like methamphetamine or cocaine. What you want is you want the dopamines to work because you need them for sex and stuff. Now this is the terrible like threat of ibogaine. As you can see, this man is sleeping on a couch. No, he's actually tripping on a couch. And they're worried about the fact that he can see stuff on the inside of those eye patches he has on. Now, as I was saying, as he said, I'm the guy who kind of like follows this REM model of ibogaine. And I just want to point out two things. You see the awake stage here and the REM stage here are almost identical. So actually, REM is in a way a form of waking sleep. And you know you can remember dreams, and if you have a really good sleep, you wake up from a very vivid dream. So REM is also like implicated in like immune health and a bunch of other stuff. So having something that like simulates REM might not be such a bad thing. Now, this is another thing that I just want you to understand very briefly, which is that this is a basic glutamate. Remember, glutamate heats things up. Acetylcholine is like cold fusion. The acetylcholine actually is implicated in somehow the transmission of light from your like eyeballs all the way down to your toes through your central nervous system, which is based on photons. And what happens is the acetylcholine is causing a steady rhythmic wave, a standing wave that like reproduces your entire environment and everything in your body image in that environment. And the glutamate is coming along like a camera and like flashing. This is infrared, and it's perturbing this. And that's how you can, like, according to this like internal clock, then produce all the other cascade of neurotransmitters. But you can't have life without like processing light, like the little microorganism here, and then backing up and then going around the rock, okay? And here is your neurons, and they're sending out nerve growth factors to each other and growing in and finding a direction like the little microorganism based on a totality of visual effects from your environment and processes based on light. So basically light turns into matter. Spirit, this is solving Descartes' problem. Spirit persuades substance. Now,
Dana, so sorry to interrupt. We are running low on time, so if you could wrap up in the next couple of minutes.
I'm at the end.
Okay,
I'm at the end.
Beautiful, beautiful.
Okay, I just want to point out that back at the, once again, back when we were doing stuff with the National Institute on Drug Abuse, we had some nice coverage. And they found out that if you give ibogaine in really big amounts, it causes some cells to be lost in the cerebellum. Well, you don't use 10 grams on a human being. It never happens. So this is never going to happen in people, and it doesn't happen in primates anyway. But what they did find out is there's these ascending bursts from the cerebellum. So you're lighting up the part of the brain where William Burroughs said the addiction is actually lodged, the part of the brain where you can like basically ride a bicycle without having to, think about it. And the only problem is when that takes over, then your feet walk you down to the dope spot and you don't even realize what you're doing until you get there to buy the cigarettes and have to pay for them. You don't know what you're doing. So this is another part of this, and this is a little bit up the brainstem. You're getting ascending bursts from the pons to the lateral genicular nucleus to the occiput, which is where you actually register stuff you see. And this is during a REM-like effect or during ibogaine, so that the prefrontal cortex can reprocess and see what's happening in the cerebellum, and you can get control of your triggers again so that you're not so much dampening your dopamine as kind of cleaning it out. And what you have, this is just coming to the end. You have the cerebellum. At first, the prefrontal cortex is running the cerebellum, but then with taking like heroin or methamphetamine, you have a shift. So the cerebellum is actually running things, and the ibogaine thing lights it up so you can have conscious control and you can start anew. Now, this is my final slide. I bet you thought I'd never get here. And I had to get rid of some stuff. But there's a group of people at Columbia called Gilgamesh. And there's a guy named Jonathan Sporn, and he may have been on this Psych 2020. And they have 100 of these ibogaine analogs. And you see, they're looking at them, which ones have GDNF and which ones are cytotoxic, the length of it, this is the one that was really promising, but really cytotoxic. But actually what we have to do, Marique, is go to the FDA, and I'm going with Talia and some other people, and we're going to try to get them to look at microdosing as, is there a range at which it doesn't even trigger the arrhythmia? And that apparently is going to be difficult to do. But I'm available to take a question or two if there is anybody that wants to ask anything. Like I said, I think that it's possible that some of these new drugs that Gilgamesh has may be better than ibogaine, if it does everything that ibogaine does. But we just didn't know anything in 1996 when they came up with 18-MC.
Mm-hmm. Beautiful. Well, Dana, thank you so much. Really appreciate it. Really interesting stuff. And I think that especially bringing in 18-MC and the work of Gilgamesh is particularly of interest. I'd love to hear your work, or your thoughts on, if there are any other companies in the space that are doing meaningful work specifically with ibogaine and taking this more broad approach of testing compounds in large
Howard Lotsof's company still exists. Mm-hmm. Howard Lotsof's company discovered the Parkinson's effect. Mm-hmm. Howard Lotsof's company is working with Amanda Fielding to resume production in India so that we can do a large clinical trial for cocaine alcohol in Brazil and be adopted by a socialized medical setup for 284 million people. I would look at Ibogrow. The previous name was Phytostan, but we're changing the name. We're getting a Delaware corporation. But if you want to work with the people who've been working on this for 40 years, who understand everything about it, everything about treatment, who only care about bringing this to the world, look us up.
Mm-hmm. Beautiful. Well, Dana, we do have to move on, but I would love for you to leave some links in the chat, just kind of sending folks to that website and to any other work you'd like to bring up to folks.
Well, there's ibogaine for the world. Ibogaine for the world is the best thing right now. Thank you so much for your time. I hope I didn't go
thank you, Dana.
much.
Thank you. You're good. Thank you so much, Dana. We really appreciate it.
Uh-huh.
Be well.